Scipion provides the tools to create the whole processing workflow in an integrative way for single particle analysis and cryo-EM to achieve a high-resolution reconstruction of the biological specimen. This framework makes it possible to create the processing workflow using several image processing packages, favoring interoperability, traceability, reproducibility, and the combination of information conveyed by different methods to generate a more accurate output. Scipion is constantly growing, including new methods and packages. Moreover, it has been extended to cryo-electron tomography and atomic modeling, allowing also to create workflows to process these data. To create a project in Scipion, click Create Project to create the project. To import the microscope movies, select Import Movies. A new window will appear. In this window, enter the path to the data and set the microscope voltage to 300 kV, the spherical aberration to 2 mm, the amplitude contrast to 0.1, the magnification rate to 50,000, the sampling rate mode to From Image, and the pixel size to 1.34 angstroms. When all of the parameters have been set, click Execute. When the method finishes, open the Summary tab and click Analyze Results. A new window will appear with the list of the outputs generated by the method. To run the optical flow method for movie alignment, open XMeep3 optical alignment method and select the imported movies as the input movies and set the frames to a line range from 2 to 13. Under additional parameters,